Right, students, good morning. So, the place where I left you out yesterday, let me give you a brief review that I have discussed so many things related to triglycerides. I have talked about all the three locations of carbon. Triglycerides is basically resultant due to the esterification reaction. It is long chain of fatty acid. If you remember glycerol, so three positions are there: CH2OH, CHOH, that was CH2OH, this is CHOH, and then CH2H again. So in the same context, you just keep on removing, H, and you keep on producing carboxylate ion, and to that, you are keeping addition of the things which are coming in, as per the reactions of triglycerides. So students, I have described about phospholipids yesterday, about phospholipids and about its type. So this thing I have told you yesterday, that what is the difference between a triglyceride and a phospholipid. If you remember, at position number 3, I have shown you out that at this location, instead of H, you are adding a phosphate molecule. Rest you can see COR1, COR2. So here you are just, instead of CH2COR3, you are observing a phosphate molecule. So I told you they are called as phospholipids, or they are also called as phosphatidate. Actually, we can say at position number 3, the hydroxyl group is esterified to inorganic phosphate. Pi represents inorganic phosphate. And in glycerophospholipid, I told you that in place of that alcohol, you are having a X. X is represented by any serine, choline, ethanolamine, glycerol, or inositol. I didn't told you inositol yesterday, but I told you that yes, it is represented by choline, serine, ethanolamine, etc. Now inositol is nothing special. You just see that X is replaced by this inositol. It is just like a glucose molecule, if you remember the beta structure of glucose, so it resembles inositol. So when an inositol is replacing that X position, it is called as phosphatidyl inositol, right? So they have, what is the role of phosphatidyl inositol? The role of phosphatidyl inositol is that apart from a membrane lipid, because it is a type of phospholipid, so it, apart from this, they are also having a role in cell signaling. Don't forget this. So cell signaling is a very important key factor over here. All right. So phosphatidylcholine is the same one as I have told you yesterday. I'm not going to repeat it again and again. I have told you how to remember it. In place of third location, you just add N triple methyl group. So you need to remember this. I'm not going to tell you everything I do every day. Okay. And yeah. So what actually happened is that, what is the polar region in this glycerophospholipid? Polar region is comprising of a glycerol, carbonyl oxygen of fatty acids, and inorganic phosphate, and this X, which is also a polar head group. So see, it is a very polar sort of molecule, the glycerophospholipid. A polar region is having, I am again repeating, glycerol, carbonyl oxygen of fatty acids, inorganic phosphate, and the polar head group, while the long chain of hydrocarbon is the non-polar end. So sphingolipids, I told you, in sphingolipids, usually the you remember one of the fatty acid and glycerol has been replaced by sphingosine. Now I have shown you the structure of sphingosine yesterday, and I told you that I will be describing about sphingosine in the upcoming classes. So you need to remember that sphingosine is somehow appearing like this. If you see sphingosine, again CH2OH at position number 2 instead of CHOH. देखो मैं हमेशा ग्लिसरॉल की बात कर रहा हूँ. Take the glycerol as a reference molecule. So CH2OH is okay. CHOH, OH की जगह NH3. You have added NH3. And in third location, CH2OH. So just remove one H. You will be adding 1, 2, and 12 and 13, 14. 12, 13, 14, 15. Right? So 15 carbon you have additionally added. So it becomes sphingosine. I'm again repeating, in glycerol, first position is still like CH2OH. Second position, in place of OH, you have added NH3, which is a polar molecule. And in third location, instead of CH2OH, 1H is replaced by this 12, 13, 14, 15 carbon molecule. This is sphingosine. So we call it as the lipids which are having a sphingosine lipid is actually, we call it as a sphingolipid. So here it is written, the derivative of sphingosine, which is a long hydrocarbon tail and a polar amino group, right? So yes, very important factor is that sometimes they are reversibly phosphorylated to produce the signal molecule, which we call it as sphingosine 1-phosphate. Sometimes you need to remember this. So what happened is that the position 1 OH has been removed by phosphate. So this is called as sphingosine 1-phosphate, right? So why? Because they have the role of in signaling molecule. So they are, I mean, derivatives of sphingosine which are found in as a constituents in biological membrane. Fine. So what happened is that the amino group of that sphingosine is actually forming an amide bond with a fatty acid carboxylic group to yield a ceramide. So you see sphingosine over here, and suddenly you see at position number 2, what happened is this is replaced. See, this RCO comes in. So I told you that the amide, I mean, it forms an amide bond with a fatty acid carboxyl. So this is a fatty acid carboxyl, right? So this structure is called as ceramide. So dekho, you started from glycerol. You know how to draw sphingosine. Sphingosine is a part of a sphingolipid. So right now I'm just telling you that sometimes the amino group of this sphingosine, I mean the position number 2 amino group, has the ability to form an amide bond with the carboxylic group of the fatty acid, right, to form ceramide, right? So what happened is that in the complex kind of sphingolipids, this polar head group is esterified to the terminal hydroxyl of the sphingosine moiety of the ceramide. So that is having a special role in cell signaling or neural transmissions. Okay, so sphingomyelin is one such new word which is I am introducing right now. I'll be discussing about this in my next uploading.